be doing with that. So welcome all of you, and Mariah, you can do everything you want from now on. So.
All right, thank you, Dean Christodoulou. Good afternoon, everybody. My name is Mariah Payochili. I'm a first-year graduate student studying chemical engineering. It's my pleasure to be the host for today's Engineering in Action speaker series on how our Center for Water and the Environment is helping to clean up wastewater. Today, we are going to hear from Center Director Carrie Howe and Associate Director Andrew Schuler. Also featured today are grad students, Derek Belka and Haley Ormsby. Before we get started, I want to mention we have a time reserved at the end of the talk for a Q&A. So I encourage you to use the chat function for your questions. It is now my pleasure to introduce Dr. Carrie Howe.
Hi, I'm Carrie Howe, and I'm the director of the Center for Water and the Environment Research Center here in the School of Engineering at UNM. The Center for Water and the Environment was created a few years ago to help develop solutions to problems with water and our environment, hence the name. And we deal with a lot of important issues that are really critical to an arid state like New Mexico. So we've got faculty and researchers and students who are investigating issues related to how forest fires and drought affect our water supply and the water that comes out of our mountain watersheds, and other faculty and students who are working on treatment technologies, treatment to treat water, make it safe for us to drink, or make water safe to put back into the environment. So things like our wastewater are not polluting our water supply. And that's what Derek's going to be talking about today. And then other topics like the interrelationship between our energy resources and our water resources. So lots of interesting and important topics related to water and the environment. The one that I mentioned today that we're going to be talking about is wastewater and how we treat it and how we make our waste streams from a municipality like Albuquerque, make that safe to put back into the environment so we're not polluting our water supply. And Derek is going to be doing the main part of our presentation today. Derek is a PhD student here at UNM. He recently was awarded a very prestigious National Science Foundation Graduate Research Fellowship, which we're really proud of him for that. I'm going to let Derek talk a little bit more about his background, and then he can jump into giving us his presentation and telling us how he's keeping our water supply from getting polluted. Derek, you can take it away.
All righty, thank you very much, Carrie. Yeah, so today the title of my presentation is Cleaning Up Our Wastewater, What Happens When Bacteria Compete for Resources. As Carrie said, my name is Derek Belka. I am a professional engineer licensed in the state of New Mexico. I'm an NSF graduate research fellow working with Dr. Andrew Schuler here at the Center for Water and the Environment. So what are we going to talk about today? We will give you a little more background on an overview of what wastewater is, for those of you who don't know, and the how and why we treat it. I'll talk to you about this really cool treatment process called enhanced biological phosphorus removal. And we'll talk about our research and how it will help design better treatment processes for the future. So my road to grad school may be somewhat atypical. It was a little bit tortuous, I guess, to get there. I actually graduated from college the first time in 2008 with a Bachelor of Arts in Political Science. And I don't know if everyone remembers what was going on in 2008, but it was not a great time to be graduating from college. The economy was not exactly booming, especially for people with liberal arts degrees. So I bounced around several different service industry jobs for a while before deciding to go back to school and get my Bachelor of Science in Civil and Environmental Engineering from the University of Washington. I then came down to Albuquerque to work at Sandia National Labs for the summer as a water power technical intern. So I was working on a project that was trying to help develop our nation's water resources in different ways to provide renewable energy to our energy grids. And then I got a job while I was here with a private municipal consultant called Moles and Corbin and Associates. Some of you may have heard of them. And then around 2020, I decided to go back to school to get my Master of Science in Environmental Engineering and ultimately my PhD. So that was kind of how I got here. Like I said, it's kind of a long, different road than I think a lot of people did. And a big part of the reason, which I'm sure you will get that flavor today, a big part of the reason I went back to do this is that I wanted to help provide solutions to environmental degradation and make sure that we're doing the most that we can to prevent degrading the environment. So let's start talking about wastewater. And I love this topic, and I will probably throw out words inadvertently that not everyone will understand. So please type into the chat box if I say something that you're like, what were you saying? I'm more than happy to answer questions. So wastewater is anything and everything we send down the drain. So that can be cooking and cleaning waste. When you clean your dishes, all that stuff goes down the drain. Hygiene or shower waste. And then what most people think about when they think of wastewater is actually fecal matter and urine. And then we also, in our wastewater, we have ground and storm water that naturally makes its way into the system of pipes underneath our communities that collect the wastewater from everyone. And what you may be somewhat surprised to learn is that that part that comes from our toilet, this is just sort of a pie chart of a somewhat typical mix of different things that go into wastewater. But the stuff that comes from our toilets, even though it's most commonly what people probably associate wastewater with, is actually a pretty small chunk. And so I think that's kind of an important thing for everyone to learn about, because there's this idea out there that someone came up with the term toilet to tap. And I really hate that, because it's like this tiny little bit of our overall wastewater mix. Okay, so these things that go into the wastewater system primarily contain suspended matter. So that's dirt, sand, things like that. We have bacteria and viruses, of course. And then we also have what we call nutrients. So that's carbon, nitrogen, and phosphorus. And these are the essential building blocks of life. And that's why we call them nutrients. Why do we treat the wastewater? Well, Carrie kind of alluded to that a little bit. In a community that collects and treats our wastewater centrally, that wastewater, well, actually anywhere that collects its wastewater, that wastewater gets discharged back in the environment. So here in the arid Southwest, sometimes that water is treated and turned around and used for irrigation and dust control and construction and things like that. We call this water reuse, which is a really important part of what we do in the Southwest. But a good portion of that makes its way back into rivers, lakes, and streams. And we want to prevent environmental degradation, because if, like here in Albuquerque, we get our drinking water out of the Rio Grande, or a pretty significant chunk of our drinking water out of the Rio Grande, anyone who's discharging wastewater upstream of us into the Rio Grande, we're now taking that into our drinking water sources. And there's this phrase or proverb out there, I don't remember who originally said it, but that an ounce of prevention is worth a pound of cure. So if we prevent the discharge of these things in the first place, then we don't have to go back after the fact and remove them before we start drinking them. And then also treating our wastewater, which is something we'll talk about this more in depth as we move through the presentation. But that if we're discharging carbon and nitrogen and phosphorus, it will naturally cause algal blooms, or the growth of algae in these water systems. And that can kill fish and other aquatic life. So if you've ever heard of the Gulf of Mexico dead zone, that's an excellent example of toxic algal blooms causing just this massive destruction of marine life. And we don't want those things to happen. I guess I should actually talk about this picture in the middle here before we move on. So this is from the 1950s, and this was taken in Cleveland, Ohio. And this is the Cuyahoga River. And there was I forget exactly what industry it was, but there was an industrial discharge of wastewater into the Cuyahoga River, and it caught on fire. And the actual river itself was on fire. And so this was one of the events that really prompted more intense environmental regulation within our country, because people realized, hey, we don't want that to happen again ever in the future. So it's just a fun fact. All right, well, how do we treat wastewater? Here in Albuquerque, we have an entity that's in charge of our water and wastewater, our water delivery and our wastewater treatment. And that's the Albuquerque-Bernalillo County Water Utility Authority. And all of our wastewater is collected and sent to what's called the Southside Water Reclamation Plant, which is this facility that's shown in the image at the right. And these, to give you a sense of scale, let me get my laser pointer out here. These are concrete structures right here. And that's probably roughly equivalent to 3 to 4 football fields. So that's a pretty massive structure. And you can see there's more of these down, and this is all providing treatment for the city of Albuquerque. And they treat somewhere in the neighborhood of 50 to 60 million gallons a day, which is this is a big facility. Not everywhere is like this. In some smaller communities, you don't have quite as large a facility, but pretty much everyone does some form of this. Okay, so what's going on at this treatment facility? We have a combination of different processes that we put together to remove those contaminants that we were talking about earlier from wastewater. So one of the methods is physically, where we actually use screens to remove large debris. We have settling tanks to let particles fall out. So that's what's shown in this picture here on the right. You can see there's kind of clear water on the top and then solids on the bottom. So we're just letting that stuff fall out of the water, and we're removing it that way. There are advanced filtration systems, but I don't have a picture of that shown here. Another method we can use is using chemical reactions to remove specific waste products. So this formula on the right is showing that we can add aluminum to the water to remove phosphorus, or vice versa, because we're forming this aluminum phosphate solid that we can settle out, just like was shown in that picture on the top right. It's another way of another version of treatment that we can do. Or we can do biological treatment. And basically what we're doing here is harnessing the preferred diet of different bacteria that like to break down and remove our waste products. And so this picture on the right is actually a microscope slide of a bunch of different bacteria growing in a wastewater system. And this biological treatment is what does a lot of the work in a wastewater treatment process. Because we have a lot of things that are in the treatment process that are dissolved that we can't necessarily remove physically, or that would be extremely costly to remove through just piecing together a whole bunch of different chemical reactions. So this biological treatment is very important to the overall wastewater treatment process and where our research is focused. So biological wastewater treatment it happens in these large concrete structures that engineers design and build. So this facility shown here on the right is actually the town of Bernalillo's wastewater treatment facility. Still probably about the size of a football field, but not quite as big as the city of Albuquerque's, because this only treats, I think about 500,000 gallons a day, so not quite as much. But it still contains a lot of the same stuff. So carbon, nitrogen, and phosphorus that are the building blocks of life, or what we call nutrients. And we remove this this process is called activated sludge. And it's kind of a fun name, but to give you an overall sense of what's going on, we have raw wastewater coming in here on the left side. It goes into an aeration basin. And in that aeration basin, we're adding air into the treatment process, and bacteria in this tank are using oxygen to eat our waste products so that they can grow more bacteria. That's basically the idea. We're just farming bacteria in this system. When we get to the next tank, the bacteria are allowed to settle out. So this actually removes a lot of the waste materials, because those bacteria are sitting there chewing on all the waste products that we have going in. They're taking it into their cells so that they can grow more bacteria. And then we settle them out of the process. We throw some of them away on the bottom. So that's this excess bacteria part of the diagram here. And we'd send some of them back into the treatment process, because we like the work that these bacteria do. We don't want to get rid of all of them. We just want to get rid of a portion of them. And this leaves behind relatively clean water, but there's still quite a bit of bacteria and viruses floating around. So we send that to what's called disinfection, where the remaining bacteria and viruses are destroyed using typically chlorine or ultraviolet light. This process is really good at removing carbon. It's really good at suspended matter removal and bacteria and virus removal, but it's not so good at nitrogen and phosphorus removal. And I'm not going to talk today about nitrogen removal, but I am going to talk about phosphorus removal. And what that looks like is this process called enhanced biological phosphorus removal. So you'll see we still have our aeration basin here. We still have our settling. We still have disinfection going on the right side. But what we added is this anaerobic treatment basin over here. And what this does, the anaerobic basin just means that we don't have any oxygen for the most part. There's also the absence of a couple other things, but then we'd be kind of getting too technical into the weeds, and I don't want to go too far out on a limb here. So basically the idea, there's no oxygen, but we keep it mixed up so that the bacteria can move around. And adding this basin causes a natural selection for bacteria that can survive well in both environments. And two of those kinds of bacteria one of those kinds of bacteria is called polyphosphate accumulating organisms. And these are the guys that remove phosphorus from the treatment process. So when they're going through the anaerobic basin and the aeration basin, they take up a bunch of phosphorus and we throw them away. And I added this little down here on the excess bacteria label that these bacteria are greater than about 5% phosphorus on average over all the bacteria, whereas normally they'd be about 1.5%. So they've taken up a lot of extra, 2 to 3 times as much phosphorus as would normally be taken up. And then there's this other group that shows up called glycogen accumulating organisms. They do not remove phosphorus, but we'll find out why they're important here in a moment. So basically, just what's going on here, we're just removing phosphorus by making these microbes take up a lot of extra phosphorus. And how does that happen? Well, in these two phases, so I've got shown here on the left and the right, an anaerobic phase and an aerobic phase. So in the anaerobic phase, we have a lot of organic carbon available. And the most commonly studied phosphorus removing organism that we have studied likes volatile fatty acids as their organic carbon source. So they need to get this into the cell somehow. And they do that by taking the phosphorus that they've stored up and the bonds, they make these big polymer chains of phosphorus or phosphate molecules. And the bonds that occur between each of these different groups are very energetic. So when the microbes actually cleave that bond apart, they get a lot of energy out of it. And they use that energy to bring the organic carbon into the cell, or at least that's one of the things they use the energy for. They also use some of that energy to turn that carbon source into a chain of carbon, a carbon storage polymer. So it's just a bunch of different carbon molecules, different types of carbon molecules chained together. So this is what happens in the anaerobic phase. But now we get to the aerobic phase, and we just released a bunch of phosphorus. And I thought, hold on, let's see. Got my laser pointer, got me messed up here. There we go. Okay, so now I just told you that we are removing phosphorus, but I said we just let a bunch of it out into solution. So we need to get rid of that phosphorus somehow. And basically it needs to come back into the cell. And the way we can do that is, well, we just built this chain of carbon, or we didn't build it, but these microbes built this chain of carbon molecule or carbon storage products. And they're going to use the oxygen that's now available in the aerobic phase to generate energy to bring that phosphorus back into the cell and create a chain of phosphorus molecules, so that when we recycle some of them back into the treatment process, they can kind of go through this all over again. And it's a really cool kind of complex metabolism that goes on in this system. And there's a lot of depth to this that I'm not showing, but this is the general idea of how we remove phosphorus. So we stress the cell in this anaerobic phase, it gets rid of the phosphorus, and then we subject it to an aerobic environment, it pulls it all back in. So just cycling through these two things causes this really cool metabolism to happen. And it sounds like we've got it under pretty good control, right? Like we understand what's going on, we can apply this and make it work at full scale. Well, it turns out that's not actually true. This treatment process has a lot of issues associated with it. And what's shown in this graph here is that in the United States, we're increasingly regulating the discharge of phosphorus from these facilities. And almost in lockstep, we see that there's more and more phosphorus violations per year. So every time we add a permit, we get more violations because people are not people, but wastewater treatment facilities really struggle with this type of treatment. And it has to do with those other organisms I mentioned, or at least one of the ideas is that it has to do with those other organisms I mentioned called the glycogen accumulating organisms. So one of the main ideas out there right now about why these facilities fail is that competition between the phosphorus removers, the PAOs, and these glycogen accumulating organisms for organic carbon, specifically volatile fatty acids or VFAs, in the anaerobic reactor leads to a breakdown in this metabolism. So in the graphic I have shown here on the left, we've got, basically this is meant to be a wastewater treatment tank. In successful enhanced biological phosphorus removal, we have the polyphosphate accumulating organisms outcompeting the glycogen accumulating organisms for organic carbon, and therefore suppressing the glycogen accumulating organism population. We get some change in the reactor conditions because wastewater treatment facilities are very dynamic. You think about it during the course of the day, you're not only just flushing the toilet, you're cleaning dishes, you're doing laundry, you're getting your car washed, all these different kinds of things that can end up going down the drain. So there's different things are coming into the wastewater treatment plant throughout the day, and it changes the conditions within these reactors. So the idea is that these changes in conditions cause an upset that allows the glycogen accumulating organisms to proliferate more than the polyphosphate accumulating organisms. And so that causes this failure of enhanced biological phosphorus removal, or at least this is one of the ideas that's out there. So it would stand to reason that if you have a significant population of GAOs in your treatment process, that you would not be able to effectively perform enhanced biological phosphorus removal. But when we look at data from full-scale treatment facilities, we see that that's not actually true. So what's shown in this graph here on the right, if you follow the green line, is basically just showing you that we have relatively stable performance of enhanced biological phosphorus removal. So we're getting good phosphorus removal, but our GAO population is increasing at the same time. So if we're getting a lot of GAOs in the system, but we still have a system that removes phosphorus, maybe they're not really the cause. And some people have suggested what's shown with this purple line is that these GAOs really only show up when we have so much carbon available that it's just not needed by the phosphorus removing organisms. They've used up all the carbon that they need. And so now these glycogen accumulating organisms have some carbon available that they can use. And so this kind of flies in the face of this commonly accepted idea that GAOs cause failures of enhanced biological phosphorus removal systems. And it suggests some other hypotheses or ideas. One, that GAOs are just opportunistic. They only show up when there's excess carbon available. It also suggests, well, maybe we've been looking at the wrong phosphorus accumulating organisms, because there are others out there. I mentioned earlier that it was just the volatile fatty acid phosphorus removers that we've really studied extensively in the past, but there are others out there. It kind of suggests, well, maybe these interactions are just way more complex than this sort of simplistic back and forth between two species or two groups. And it also suggests that if we design our systems to sort of be less selective, have more different kinds of organisms in them, that they may be more resilient to changes or those dynamic reactor conditions that may cause process upsets. And I think this kind of segues me into this, going back to that second hypothesis, that other PAOs may be important contributors to enhanced biological phosphorus removal. There was a really cool study that came out a few years ago that showed that this group of volatile fatty acid users that remove phosphorus from the system, so that's this group right here in the white in this graph, they're the ones that use volatile fatty acid that we've studied extensively in the past. And then there's this other group of amino acid users, or at least that's what we think they use. that show up in greater abundance and also appear to account for more phosphorus removal. So that's kind of an interesting thing, right? Like we've been studying this one group for a really long time, and then it turns out, well, hey, maybe they're not really the ones doing all the work. So I think, let me get this out of the way again, sorry. It shows us that, well, VFA users remove less phosphorus than amino acid users, and that's a pretty important finding. It also shows us that amino acid metabolism could influence how well nitrogen is removed, because amino acids are chains of carbon with a nitrogen group on them. And it also, or one of the other questions we've been trying to figure out is how does this amino acid metabolism use phosphorus? Is it using it in the same way as the volatile fatty acid phosphorus removers? Because that could be really important to how we design our facilities. So I'm going to kind of walk you through the current idea of how this metabolism differs from the one we looked at earlier. So instead of volatile fatty acids out in solution, we now have amino acids out in solution. And we want to get those into the cell somehow. And we think, at least right now, what we're trying to figure out is that basically these microbes are doing the same thing that the volatile fatty acid microbes are doing. So they're cleaving off phosphate, they're getting energy from that bond to bring amino acids into the cell. But something that's kind of interesting is we don't detect that same chain of carbon stored within the cell. At least there's one research or a couple research papers out there that seem to suggest that they just store the amino acids just kind of loosely within the cell. They're not binding them up in anything. So then when we get to the aerobic phase and we need to get phosphate back into the cell to create these chains of phosphorus for use back when we get back to the anaerobic, we now have these amino acids loose within the cell that are available for metabolism with oxygen to provide energy, but they release ammonia when they do this. And at least we've got some preliminary data that suggests that they release ammonia. So that's why I was saying it's kind of important to the overall balance of also removing nitrogen, because now we've presumably added or converted the form of nitrogen within this system, which is an important thing for the removal of nitrogen. So our work in the lab to kind of investigate this new exciting metabolism that we don't know a lot about is we're actually growing these pee removing organisms on one group or in one of these reactors. We're growing the pee removers on volatile fatty acids. In the other reactor, we're growing them on amino acids, so that we can try to ask some questions or design some experiments to answer some questions. One of those questions is, what conditions do amino acid phosphorus removers prefer? Does this differ from the volatile fatty acid phosphorus removers? And if so, how and why? We're also trying to figure out what conditions cause these microorganisms to fail. So is it the pH? Is it the temperature? Is there not enough carbon available? What causes their populations to crash, so that we can try to design better systems that don't fail as frequently? And we also want to know if failure is really caused by GAO competition. So if it is, that's something that's going to be really hard to control in a wastewater system that's inherently dynamic and always changing. Like, how can we control a certain population? So that's a whole different thing that we may need to get into in order to really make these systems robust and reliable. And this all feeds into this question that I've said so many times now: can we design systems to do a better job to prevent these breakdowns in phosphorus removal, so that we have more reliable systems? We're not causing algae blooms. We're not poisoning our water supplies. Essentially, we want to make sure that these systems are reliable, so that as we continue to regulate the discharge of phosphorus from our wastewater treatment facilities, we can do so more reliably. There's some good references here if you really want to read into the weeds of what I've been talking about. And I just want to say thank you very much. I know that sitting through another Zoom meeting in the middle of the summer, when we're about ready to be liberated from Zoom for the foreseeable future, I really appreciate you all coming today. I think we wanted to wait and do questions a little bit later, but we could do some questions now because we're going to have a short break while we transition into the lab, because I want to show everyone like how are we doing this research in the lab. It's nice to look at pictures, and I'm sure you're done listening to me drone on. So it'd be cool to see what we actually do in the lab to investigate these questions. I will stop sharing.
Yeah, so don't forget to use the chat function to bring up any questions you might have. I think we had a few starter questions, so I can just bring up one of those. What is the most helpful thing we can do to keep water clean?
Ooh, that is a very loaded question. The most helpful thing we can do to keep water clean. Be mindful of what you put down the drain. You know, if there's something that you think might be toxic, there's tons of great resources out there that tell you, is this safe or not to put down the drain? Usually the utilities website will have a link to that, or the EPA, Environmental Protection Agency. But also I think just being mindful of any potential chemical you can put down the drain. Or even on you if you're gonna go swimming in the water, so like sunscreen. Even though you wanna have it on you, if you have sunscreen on, you go swimming in a lake, you're leaving behind that sunscreen, right?
Yeah. Okay, cool.
Just to add a little bit to what Derek is saying, some examples is, so around town, you might see little plaques by storm drains in the street that say, don't dump oil here. So, you know, in the past, people used to change the oil in their car and then think, oh, I'll just dump it down the storm drain, you know, because it goes away. I don't have to deal with it. Well, it goes to our river and it pollutes our river. So that's one example of, you know, dumping waste into our water supply. And then another example that's becoming popular, people used to be told to discard all their leftover pharmaceuticals, their leftover medications, flush those down the toilet. Well, that's not a good thing to do with it, because that ends up in our water supply. So now you'll see that there's medication take-back programs. You should take them back to the pharmacy, or possibly if it's a controlled substance, it needs to be taken back to a police station. But those are some examples.
Great, thank you. So I think you wanted to transition to the lab. I don't know if we want to continue with that, and then we can answer some more questions after that if you'd like to, Derek.
Yeah, I'm trying to get my my cell phone to switch audio over here. So if anyone needs to, you know, go get some water or something, now would be a good time. Oh, I think, can someone make the second me in here a co-host? I think that's the problem.
I don't think I have the ability to do that. I would.
I'm sorry, say that again? What do we need?
Oh, sorry. Yeah, Derek, his other account, if he could become a.
Oh, yes. Derek, what's the name of your other account?
It's me standing there in front next to a big valve.
All right, how about that?
Derek, I could probably use my audio if you're the one recording. I can talk to my laptop.
Okay, we're going to transition over to the lab. I'll be right back. I got to get on my proper PPE to be in the laboratory, so I'll be just a minute.
So while we're waiting for Derek, are there other questions that I can answer? Let's look at the chat. Someone asked, do we recommend drinking tap water? Yes, we do. So you've obviously familiar with there's various sources of water available, tap water or bottled water. Well, when you think about it, all water comes from the environment someplace. So bottled water is coming from the same places that tap water comes from. Both have to come out of the environment. So they're coming from a lake or a stream or a aquifer someplace. They have to be treated and made safe to drink and then delivered to you. And so you can have water delivered to you through pipes underground that go straight to your house, or you can have that water put into bottles and shipped to you in trucks. And the bottled water costs anywhere from 100 or 1,000 times as much. When you think about it, you're paying fractions of a penny for a gallon of tap water, and maybe $1 or more for the same amount of bottled water. And it's not necessarily better quality, and there's a much greater environmental impact associated with all that bottling and all that trucking. Let's see, another question I see here. With the river so low, how do we keep the tap water flowing? So the water authority here in Albuquerque has actually a very sophisticated system to make sure that we have a reliable water supply. And it relies not only on water in the river, but also groundwater in the aquifer beneath Albuquerque. And so the water authority here uses water from the river when it's available and uses groundwater when there's less water available from the river. And there's different types of treatment that are provided depending on where the water is coming from. And so our water authority here has a very detailed plan to provide a reliable water supply into the future using these two water sources. Let's see, other questions, or unless Derek is ready now.
I think we're ready to go, and my phone appears to be working again. So.
Okay. So can we make that screen larger? So I see a screen that has Derek showing the lab. Can we somehow make that the main screen?
Let's see, yes. Let's see how I could do this.
There's a thing that says spotlight for everyone.
Yeah. Okay, cool. Okay, so we are now in the laboratory, and I'm going to turn this over to my laboratory assistant, Haley Ormsby. And is it going through your headphones now? Okay.
Yeah, I'm hearing an echo.
Okay.
Okay, hello everyone. My name is Haley Ormsby, and I am Derek's undergraduate research assistant here at CWE. I started working for him. I was an undergrad, but I recently just graduated with my bachelor's degree, and I'm working on my master's right now. Or I will start working on it. So these are our reactors here. Like Derek said, one of the major applications of wastewater treatment is enhanced biological phosphorus removal. So to get an idea of how our reactors are working and how the microbes in our reactors are performing, we have to do tests, including phosphorus testing, to see how much phosphorus is being released into the net during each phase of the reactor. So to do that, we have these streamlined kits from a company called Hawk. They look like this, and they just come with pre-made vials with reagents, either in the vials or they come in like powder pockets. So one test that we do is a soluble reactive phosphate test, and phosphorus doesn't exist by itself as an element in water. It's always in a compound with something. So what these tests do is they convert the phosphorus compounds into something called orthophosphate, which is basically just a phosphate ion. And we can measure that directly. So these tests are a colorimetric test, and it combines orthophosphate with some reagent, and it turns the solution yellow. And we put these vials into an instrument called a spectrophotometer, and the spectrophotometer shoots lights through the vials and measures how much light is being absorbed by the solution. So we can take that absorbance value and relate that to the concentration of phosphate in our samples. Another test that we do is we do a total phosphate. So if we want to see how much particulate phosphate is in our reactors, then we have to digest it. So first of all, organic phosphate is very hard to break apart. So we have to first digest it with heat and sulfuric acid, and then also include a strong oxidant, such as potassium persulfate, to break apart those phosphate ions, and so that we can measure that. Yeah, so that's total phosphate. We also do an ammonia test where it turns the vials green. And like Derek said, the metabolism of ammonia is not quite clear, so that's one of the goals of this research. So this is just a typical day in the lab, just taking samples from the reactors, doing different testing. We waste from the reactors every day. And yeah, we do TSS, BSS measurements. So it's total soluble, total suspended solids, and volatile suspended solids. So the TSS gives you the concentration of the total suspended solid in an activated sludge sample, and then the VSS gives you the concentration of the organics in the activated sludge sample. So yeah, that's pretty much a day here in the lab. And I don't know what else to say. Yeah, it's been fun working with you, even though it's kind of smelly.
Let's see, Carrie, perhaps you might want to field any other questions. Thank you guys for sharing that.
So Derek, I think you were also going to maybe show some images from the microscope. Is that right?
Yeah, so we're going to walk back across the hall now. And so what Haley was talking about, we have to, as part of operating these reactors, we have to characterize what's in them. We can measure the phosphorus release and uptake, and that tells us how active these microbes are. But in order to tell what's actually in the wastewater reactor, like what specific microbes are in there, we use microscopy. And so we have, you may have noticed behind me in the lab earlier, we had this microscope sitting against the wall over here. And give me 1 second here. I'm going to stop my video on this guy while I get the pictures up. But yeah, so we want to, once we've characterized what's actually happening in terms of phosphorus removal and uptake, we want to look at what's in there. And we can use this really cool tool called fluorescent in situ hybridization, or FISH for short. And what this does is we actually, we attach a genetic marker to these microbes. And so what's shown here is sort of the general, it might be kind of hard to see through the phone. This is an old computer, so it's difficult to get. But see, there's like individual microbes there that are labeled with this green fluorescent tag. And then if we use a different genetic tag, we can get a different set of microbes. So actually, this is a better set of images here. So here, this green one is showing, this is all the bacteria that's in the wastewater reactor. And then we flip, and this orange is just the microbes that are known to accumulate phosphorus. So we've got the full spectrum of everybody in the green, and then just the orange ones. And this is actually from a reactor that was pretty much completely phosphorus removers. So that's why the majority of it is orange. It's a pretty cool tool. The microscope does a lot of work for us here in the lab.
All right, well, thank you again for sharing your research with us, you know, and everything that you're doing to keep our water clean. So I think now we'll start the question and answer portion. So as most of you have done, just drop them into the chat. I think we had one from Steffi earlier. She asks, as a person who has a well and lives with people with septic systems, how does nature do all this in groundwater?
Sorry, I was transitioning my audio. Can we repeat the question?
Sure, yeah. As a person who has a well and lives with people with septic systems, how does nature do all this in groundwater?
Yeah, so usually there are development ordinances that prevent you from drilling your well too close to a septic system, but of course that doesn't always happen. But part of what goes on is in a septic system, you're sending that wastewater out into the soil, and the soil, especially in the Southwest, a lot of it actually evaporates up and out through the soil. But there is a certain level of like natural microbial treatment that occurs in these systems. If you happen to be like really close to water level, which is kind of unusual. So there's also a pretty big separation between your septic system, which is maybe a few feet below the surface, and your well, which could be anywhere from 50 to 100 or even more feet below the surface before you get to the water that your well is actually accessing. So that's a really good question to ask. And it varies from place to place, but usually the discharge from your septic system does not ever make it to the same depth as your groundwater, usually. And the soil provides a certain level of natural treatment. One of the things also is that phosphorus is pretty reactive with metals. So natural metals that occur in your soil will also tend to absorb the phosphorus that comes out in that system.
Cool. Thank you. So Jens asks, with the river so low, how do we keep the tap water flowing?
That is a great question. One of the ways we can do that is by reducing our water use through water conservation. So that's things like low flow toilets, high efficiency washing machines, and generally just engaging in other water conservation practices like converting your, if you have a green grass lawn to a xeriscape lawn, which uses a lot less water. One of the other things that we do that the Water Utility Authority is starting to work on expanding here in the Albuquerque area is water reuse. So we take the highly treated wastewater from the wastewater plant and it gets sent back out into the community for irrigation. So when you see, usually when you see like a park being irrigated, that's potable water that's being used for that purpose a lot of time, or water derived from the drinking water system. And so by replacing that with treated water from the wastewater plant, which is of more than sufficient quality to irrigate a park or something like that, you can cut down on the fresh water that you need to take out. So that's one of the big components of, I think Carrie was talking about the Water Utility Authority's Water 2120 plan, which is their long range planning document for water, water in the Albuquerque Valley or down here in the Rio Grande Valley.
Okay, great. Let's see, I think that one might have been answered. So we have one from Jens. What's your origin of the wastewater that you are testing?
So I took samples from the Albuquerque wastewater treatment plant, and that was used to seed the reactors that I showed it that we had in the lab there. So it started out as basically just wastewater straight from our community. It has since morphed because we're feeding it with something that is roughly equivalent to a wastewater source, but is obviously not the exact same as raw wastewater coming through our pipes. So We make up every day, or that the reactors in the lab run on a 6-hour cycle, and we feed them with a carbon solution and a solution that has nitrogen and phosphorus and magnesium and calcium and a bunch of other different components that kind of simulate that wastewater.
Okay, cool. Another question. What information do you have about gray water use? Are there any systems?
Yeah, so I don't know how common gray water use is in New Mexico in particular, but I know that there are different ways to do that, right? So you can use gray water outside of the home, but I don't know if you've ever heard of the earth ships up near Taos, New Mexico. They actually they collect their gray water and then they use it for flushing the toilet. Okay, thanks, Bill. Balloon Fiesta Park uses gray water. So yeah, I mean, there are opportunities to use gray water. Usually the problem is that you need a completely separate pipe system to manage it and move it from place to place. And so that's one of the issues. That's one of the reasons why it's not more widely done, but it can be used in what I would call a decentralized way, where it's like the gray water is generated right on this site and used right on this site.
All right, we have another one. Can we still use our wastewater with dish soap to water anything?
Well, that depends on what kind of dish soap you're using and how much dish soap you're putting in. So if you've got, you know, mostly dish soap in the water, maybe not, but if it's just like a little bit left over, it's probably not going to be it's probably not going to be terrible. But you know, you can always keep an eye on your plants, and if they start to droop or wilt, then that's kind of a clue that maybe that's not working out so well.
Okay. We have one from Nada. How is fracking affecting our water resources?
That is an excellent question. I will admit that that is a little bit out of my range. What I do know is that there's some really interesting research going on right now about how to use water more efficiently out in the oil fields. And I believe New Mexico State has a pretty big partnership with Bureau of Reclamation and someone else. And I don't think UNM is part of that, but there's a lot of really interesting research out there as to how we prevent that from becoming a big issue.
All right. We have somebody interested in visiting your lab. Is that possible? They said.
It can be made possible. I think the university is technically opening in August, but I don't know what the policy is on visitation for non-students yet. It would be more of a question for like the admissions department, maybe. They might have a better idea on whether that can be sort of hosted, or I don't know, Carrie, maybe you know if that's like something we can do.
I think we're coming out of the COVID restrictions. We're kind of in progress right now, and I think we're going to see. I think that's going to be possible, if not in July, possibly in August, for you know, someone just from the public to be able to visit our labs again. So what I would suggest is whoever asked that question, send an email to Derek or to me, and we will you know, kind of assess where we are in terms of COVID and access restrictions, and we will arrange a time that you can do that.
Great. It looks like you could also contact Toby Tafoya. He had put that in the chat. But I think we have time for one more question. Caitlin asks, what kind of infrastructure changes, physical or otherwise, would need to be made at wastewater treatment facilities to optimize phosphorus treatment based on your research findings?
Yeah, so that's a great question that we're still working on the answer for. But I think one of the things that's come to light in the last couple of years is that focusing solely on volatile fatty acid using phosphorus removers as the microorganisms that we want to do the most work is less than ideal. So one of the ways that they're specifically selected in treatment facilities is something called primary fermentation, where we ferment some of the wastewater on the way into the treatment process. So rather than having it in a big mixed reactor, we just let it sit in this kind of quiet basin. And that actually results in the production of volatile fatty acids. So there's a couple of different ways that we can do it. One of the ways could just be eliminating that step and just having sort of a different layout of our treatment system, but there's also benefits of having primary fermentation. So that's something that we're going to try to explore of like, okay, now that we've got this information on how this metabolism works better, which is something that we're in the process of developing, then we can start working on optimization solutions of like, do we need to completely reconfigure these treatment facilities to operate in a different mode than what they've been configured to operate in?
Okay, great. Thank you for answering that. So I just want to say thank you to all of you again for sharing your research. And also thank you to all of the guests here. Our next Engineering in Action series will be in the fall. So we look forward to seeing you then. As always, stay safe and go Lobos.